Mikala Egeblad is coming from Cold Spring Harbor Laboratory in New York. And Mikala started her career in Copenhagen in Denmark, where she got her bachelor degree and master's in the University of Copenhagen. And then she did her PhD in the apoptosis lab, working on ERB receptors. And then she moved for her postdoctoral training to UCSF, the lab of late Dina Verb. And she started developing there together with Andy Ewald intravital imaging methodologies to visualize real-time tumor stroma interactions, working on the PY middle T model. And this is the time where I was lucky to actually meet Mikala. We've known each other for quite some time, I guess it's been. And in 2009, Mikala started her lab in Cold Spring Harbor Laboratory. And just this year, she was promoted to full professor. I'm not going to go through her awards and the amazing grants that she got, but I'm just going to mention that her seminal works on the NETs, neutrophil extracellular traps, have been in the last 5 years really changing the field. And in the last year, she actually started unveiling how NETs can contribute to COVID damage in the organs. So without further ado, Mikala, take it away.
Thank you very much. And thank you first to the organizers for this invitation. When I accepted it, I had really thought we would be in France and enjoying being there all together. But yeah, here we are, zooming away. So I'll talk about tumor stroma interactions in metastasis, many different types, and mostly give you a little bit of examples from our different studies on how we are implementing imaging into our studies, but also in the end, I'll show you a new imaging window that we have developed and where we are happy to share the protocol. So we focus on metastatic cancer because it is the worst type of cancer. If you look at 5-year survival for localized cancer, for most cancers, it's decent, but as soon as you have a distant metastasis, it drops down. Even for something like breast cancer and prostate cancer, the 5-year survival really drops down. So we recognize that metastasis is a very complex multi-step process that involves interaction between the cancer cells and the host, from invasion, intravasation, the survival in the circulation, extravasation at the distant sites, the formation of micrometastasis, and finally the growing of the metastasis at the secondary site. We have decided to focus mostly on the second part at the secondary site. And the reason for that is that when a cancer patient is diagnosed with cancer, in many cases, this dissemination step has already occurred. And so if you want to have a major impact on survival, we feel we need to work on this area. The first work we did on this was started with lung imaging, and it truly started with imaging experiments. We were inspired by work from Max Krummel's lab and adapted his window technology over the lung of mice. You can see here they're intubated. And then we took a classical but very aggressive breast cancer model called the 41 cancer cells. They are labeled here with RFP. And you can see after injecting them that they launch here in the lung tissue. You see neutrophils around them. And then we had injected the mice with a DAPI stain, really just to get counter stain for the lung tissue. But notice this cloud of DNA developing around some of the cancer cells. So of course, the first thought you have is that this could be just the cancer cells that are dying. But when we use the sister cell line 427 that is not metastatic and is indeed dying in large numbers, we don't see a lot of these clouds. And furthermore, when we used a probe, commercial probe for neutrophil elastase activity, you see the probe active around these clouds regions, but not around any smear of DNA you might find in the 4T01 cells. And that we have quantified here. This was scored blinded by 2 independent observers. So really, when you see most of these clouds in 4T1 cells, after 41 cell injection, it is associated with neutrophil elastase activity. So what are these DNA clouds with neutrophil elastase activity? So we learned that they are something called neutrophil extracellular traps or nets. So neutrophils are one of our major inflammatory immune cells. They can take care of pathogens by phagocytosis. They can degranulate, or then in 2004, it was discovered that they can form these neutrophil extracellular traps, NET or nets. They do so through a process that involves an enzyme called PAT4 that citrulates histones, leading to chromosome decondensation, and then enzymes associated with the DNA, neutrophil elastase and MPO, which are normally found in the granuli, transverse into the nucleus and are associated with the DNA. This is expelled, and we like to think of this like a Spider-Man that is casting a web, toxic web, to capture the bad guys. In the case of our cancer cells, there was no infection, but what you can see here in vitro cultures was that we discovered that when you co-culture the neutrophils with the 41 cancer cells, they form these nets. And here you are seeing in scanning electron microscopy the DNA strings. We could also find that it's not all cancer cells that will do this. We found that it was the most aggressive metastatic cells that would do so, but non-aggressive cells like the 4T07 would not. These DNA strings or the nets had an effect, and we showed that by using DNA-coated nanoparticles to dissolve them inside the mice. And you can see here that you're reducing the metastatic spread in the 41 mouse model as quantified over here. This is now taken on into a larger field. This is an example from another group at MD Anderson that found that primary ovarian tumors can also trigger this net formation distally. So they secrete cytokines that recruit neutrophils. Some of these cytokines are also inducing the neutrophils to form nets. GCSF was the one we found in our model, and they found it also in theirs. Then there is a formation of a pre-metastatic niche that make it easier for the cancer cell to launch. And in addition, it's also thought that the nets can function by entrapment of the cancer cells. And we are finding also that the protease activity is remodeling the matrix to help the cancer cells grow and adhere. You can inhibit it, and that reduces metastasis. Now, this is sort of the first steps of metastatic cascade getting into the tissue, but we were also very interested in understanding the step from dormancy, the step from a single cell to a growing lesion. And in case of breast cancer, sometimes this can take years. So what is regulating this step, and why do they stay dormant or latent so long? So a new postdoc at the time in my lab, Jean Elbringis, joined, and he reasoned that with nets could be induced by the cancer cell, the metastatic aggressive cancer cells, to promote metastasis. Maybe nets that were externally induced, such as in the case of inflammation, could affect these dormant, less aggressive cells. So the model he set up involved stimulation with lipopolysaccharide, or LPS, also known as endotoxin. And so LPS was then injected intranasally in the mice. And then another postdoc in the lab, Mario Shields, then did the imaging here in the lungs. Now we have changed to these 3D printed lung imaging windows. But otherwise, it's the same procedure as Max Krummel had reported. You can see here in the context of mice that have just been injected with PBS, you see neutrophils around the alveoli. These are the dark areas where the air exchange. And then we have injected in this mouse a neutrophil elastase activity probe, but there's not much activity, as you see. This is a mouse where the mouse received the LPS 16 hours before, and you see a lot of infiltration of neutrophils. They also flatten out and becoming activated, and you see the neutrophil elastase activity probe. So then we combine this with the Fouché cell cycle reporter. So this is reporter many of you are probably familiar with, where the cancer cells, or any cell, if you use it, will change color from green to red to yellow as they go through the cell cycle. In G0 and G1, they will be red. So we engineered our cancer cells, a dormant cell line, to express this reporter, injected it into mice, and then stimulated with LPS 3 times over 6 days, and did long analysis. You can see here in these stills that at day 21, they're single cells, they are red, so they're G0, G1. We think they're G0. And then at day 11, 12, 13, and 21, after LPS injections, so starting here, you can start to see that the cancer cells are switching colors from red to yellow, and you start having these proliferating mixed colored lesions. And this is quantified here. So really, in the case of PBS, we did not find any cells that spontaneously switched. So we used this system together with the neutrophil elastase probe to try to see if there was any correlation between the areas where they were neutrophil activity and where they switched. Again, this is a PBS treated mouse where you can see the red cell. And over here, you have a mouse that is treated with LPS, and you can see we have already a cell that has started to switch. We have a red and a green right next to each other. And this is found in this area of neutrophil elastase activity. Now, more recently, we've become really interested in the idea of the metastatic microenvironment playing opposing roles depending on what kind of activities is activated. So what I've shown you, and there's been a vast amount of literature on this, is that the metastatic niche, either pre or pro, either before the tumor gets there or when the tumor gets there, can be very pro-metastatic, and that in many cases involve immune cells, particularly of the myeloid derived lineage. But of course, there's also a lot of literature suggesting that, strongly proposing that the metastasis can be restrained by the immune system, particularly the adaptive immune system. For example, it's classically noted that organ transplantation from cancer survivors combined with the immune suppressive therapy in the recipient sometimes led to outgrowth of these dormant latent metastases in the recipient. So some of the work along these lines was done by my neighbor, Douglas Farron, here at Cold Spring Harbor, and he developed this model that we now are adapting, where a mouse is, so to speak, pre-immunized with the cancer cell. So the idea is that in a cancer patient, the immune system is trained to see the cancer cells and the neoantigens that may be developing. So you want to train the mouse immune system to do the same, to truly model the metastatic niche. So then one can eliminate the tumor cells. We are now doing it with surgery, but he did it with a more pharmacological approach. And then you can inject new cells, bypassing this cell, but this normal dissemination step, but into a mouse that's ready. So you know that the new cells is coming from the ones you're injecting. So if you do that, you can see naive mice modeling how we normally do metastasis assays. There is a growth of the metastatic lesions. This is in the liver, followed with bioluminescence. But in the pre-humanized mouse, you can see that we really drop below the detection level. When you look in the livers, it's not like there's no cancer cells. They're actually found as single latent cells, and they're not Ki67 positive. So we used imaging to look at this, speculating that probably it would be the adaptive immune system that would be trained. And indeed, you can see here in a mouse, a couple of days after injection of the pancreatic cancer cells expressing blue fluorescent protein, that we have around some of these cancer cells a very strong T cell response. But others, and we are presuming these are the ones that are developing into the latent lesions, that these have no recognition by T cells as far as we can see. And of course, what we are really interested in now is understanding why was these recognized, why are these not recognized, what are the signals? One thing we know from the study done by Dr. Ferrand's group was that if you deplete T cells at a later stage in these mice, these cancer cells will spontaneously grow out, a little bit less so in the later stage. And of course, we want to try to understand this difference. Is it just that they died over time, or have they reached into a deeper level of dormancy, so to speak? So to get into that, we need some process where we can follow a lesion over a longer time. And to do so, classically, we have wanted to use imaging windows, as they provide long-term access and for repeated measurements at the same tissue site. The classical model was this one with the skin flap model from Rakesh Jain's lab. Also, brain imaging windows were developed very rapidly. And then Jaco van Reenen's lab and others have developed these ring windows, which Jaco taught us a couple of years ago. So you can see here, we've been trying to implement them in breast and in the liver metastasis. And we did get data out of it, but I have to say we had some problems with them too. Some of the problems was that sometimes the mice would rip them out. We would occasionally get breakage of the glass, which is obviously not what you want. And we also felt that the glass itself could be quite inflammatory to the tissue, and we would sometimes see an inflammatory response that we suspected had to do with the stiffness of the glass rather than anything else. And of course, that's a problem if you're studying an inflammatory response. So two graduate students in my lab, Laura Majorano and Margaret Shevick, got the idea of using a silicone polymer instead. It's very easy, actually. So you mix it 1 to 20, you degas it, and you cure it at 80 degrees. And this is a kind of similar material as you're using in contact lenses, which I'm wearing right now. So this is known to be safe and relatively stable. It had already been reported, and this was the inspiration for us, in a paper in Scientific Reports, that this material could be used as a brain imaging window. And so we developed these things. You can see here that it is totally easy to look through. And we've been developing this over the last couple of years and just submitted the protocol. And I want to bring to your attention that in parallel, a very similar approach has also been submitted to bioRxiv, where you can see this different approach. So it's a very similar material and idea, but they are inserting the window slightly different than we are. The way we are inserting it is mostly using glue. We're not using glue all over the surface, but here you can see opening for the liver. We're opening peritoneum. Then making sure that the liver is easy access to the window. And then you're inserting the window first around the liver with the glue around, in a circle around. Then you push it to the glue here, push it under the skin, and then glue it together between the skin and the peritoneum. So this way, we are finding that we can get very stable long-term imaging in the mice. We've done this here, showing it for the liver, but we've also done it for the pancreas and for the mammary gland with almost similar results. So you can see here that in the mice, it doesn't look, you can barely see that there's a window, and here it is after 35 days. So again, it looks very stable. This is what the mouse look like with these windows. So they don't seem to mind it much. We've also measured their weight and inflammatory markers, and there doesn't seem to be much effect on them other than the very acute effect of having surgical implantation. But they move around very normally with this. We used our myeloid marker reporter mice to look at whether there was any infiltration of inflammatory cells to the surface, the problem we had with those glass windows. And we're seeing very little difference over time here, shown for the first 5 days in the liver. We also did histology with a pathologist, Erby Wilkinson. And what you're seeing here is that there is regions on the surface of the liver, as outlined here in red, where you're seeing granulation tissue, as they call it. And you can see it here in higher magnification. So it's not completely inert. And we think that but it's very localized, and we think it's where the glue is actually touching the liver. And so what you have to do, of course, is finding an adhesioned area, which is fairly normal and where there's no glue. One other thing we've done is we have embedded these stainless steel grits into the silicone polymer. And so by doing that, we now can simply count how many of these grits we're going into to find the same location, and then follow over time. This is shown here over a mammary gland, and you can see through the eyepieces it's very easy to find these grits and find the same location again. The first ones we were using were actually stolen, so to speak, from electron microscopy, but now we are getting them cut in the size that works for us for the opening here. So this is an example of some of the things you can see. We have here at day 2 and day 4 in the mammary gland found the same location. This is a project that's still ongoing, where we've been looking at tissue remodeling in normal physiology, namely the involuting mammary gland. So the mice become pregnant, the mammary epithelium grows, they start lactating, and then when you wean the mice, now the mammary gland will start involute and shrink back again. And this is a very dramatic epithelial remodeling process. The epithelial cells will undergo apoptosis, and there will be a lot of inflammatory cells coming in, including neutrophils. So as you can see here, we can follow the influx of the neutrophils over time. And here in the later time point at day 4, you can see that we have more neutrophils at this point. We also have a higher number of them that are expressing the neutrophil, that have active neutrophil elastase activity. And you'll also know that there is indeed a decrease in the density of the epithelial cells marked in with CFP expression. Of course, now we are implementing this to the metastasis question. This is something that is been delayed by COVID-19, and one of the issues we had was we couldn't get our microscope service during that whole time. But hopefully that will happen within the next 2 weeks as everybody's getting vaccinated. So here you can see an example of the grid over the liver, and the following of a single cell that arrives to the liver metastatic to form a metastatic niche and is growing over time and start to spreading. And of course, now the key is following this over time and see which immune cells are coming in and what are their activity levels. So with that, I will conclude by just summarizing that we are currently focusing on using intravital imaging to looking at the later stage of the metastatic cascade, the early colonization. How do the cells get in? How do they survive when they first are getting there? And how do they escape the immune control to become an actively growing metastasis? These are the main questions ongoing in my lab. And I want to thank the people in the, they will not do this. I wanted to thank the people in my lab that did this, particularly Laura Mariano and Margaret Shevick that got the new windows to work, as well as Mario Shields and Bobby Wasagi that did imaging in the lung originally. And with that, I'll just add this quote from 2002 with some of the early intravital imaging. I really find it extremely useful to look at what is happening, and then from there form hypotheses that we can test through imaging and other means. So thank you very much for your attention.
Thank you, Mikala. That was wonderful. So we have a couple of questions, and Florian, how much time do we have, considering it's 9:30 right now?
No, it's okay. Just yeah.
5 minutes is fine. So from Takashi Nishina, he's saying bone metastasis also occurs in breast cancer, and do you know if NETs are also involved in the bone metastasis?
It's a good question. So we have some fluctuation in our model, and if initially we saw no bone metastasis, then there was a period where we saw a lot of bone metastasis, and now we are not seeing any bone metastasis. I don't know if it's a microbiota of the mice or if it's a LPS batch or what's going on. there was a study that came out in Nature last year where they were looking at NETs in humans, and they were really based on clinical data, promoting the view that it's most important in the liver and the lung. So until otherwise proven, I think I'll go with that the lung and liver is probably where it's most relevant.
Okay. So another question about the nets. Do they only supply mechanical protection to cancer cells, or do you know any signaling that's going on?
Yeah, so they have a variety of different functions. So they have the mechanical support. They have the proteases on them that is altering the extracellular matrix. It's likely also altering some cytokines. They have histones on them, and histones can be toxic for certain cells, particularly endothelial cells. So they may indeed damage the endothelium that way. They can trigger platelets to form clots, and that can also be relevant in cancer and has been shown to be relevant. And then we are finding, of course, that they can affect other immune cells. That's one of the things we are focusing on right now, how they are activating macrophages and T cells. Some of that activation involves probably the DNA itself. As you know, there are DNA sensors in many cells, and so that's one of the things we are taking apart. There's a lot of literature showing that can activate the inflammasome pathway, and we have totally confirmed that. And then finally, on the cancer cells, there is a receptor called CCDC25 that was identified in the study I just mentioned in Nature last year. We are looking into that too.
Okay, thank you. And from Maria Loela, she's saying, how do you immobilize the mice with these new windows? Is there any different way than?
No, so it's mostly done after the fact by the software. So we are using the alignment of the images rather than immobilizing the mouse. Mostly we just immobilize the mouse with, lab tape. But we can share the software algorithms. A new postdoc in my lab made those.
Okay, so they're similar to the jitter corrections in ImageJ that we have. Yes. Okay. Okay.
And then the other thing is we are using a trigger, respiratory trigger, to take the images at the same time in the respiratory cycle. That really helped.
Okay, and Jackie saying we're definitely interested in collaborating with the new intravital window. So, well, it's coming up in Jove soon. So with all the details. And thank you so much. We're going to continue the discussion after all the talks are done, and I'm going to hand it over to Antonio.
Thank you.